Before I get started, I did want to mention a few things personally. First, for those of you that have met my mother-in-law, Rosa, she is back home in San Diego with us and sends you her best wishes and love at age 86. It's a little challenging to be traveling across the pond, so she decided to stay home this time, but she wants you to know that her thoughts are with you and loves all of you like all of us. So that's number one. Number two, first of all, I really want to say what an incredible honor and privilege it is for me to be part of this group, the American group, the Spain group. It's been really incredible to see, dating back to 1998, how these groups have grown. I think all of them are great, and I think they all complement each other really, really nicely. But what is so important is the quality of the people, starting with the leadership. So Gisa and Camille, I know Gisa had to step out. Camille, I don't know if you're here somewhere, but there you are. You guys have done an incredible job. And I know the husbands are in the background, but it's really the women that rock here. So just like the group in the United States, you saw Lindsay and Jessica the other night, and they've been involved something like 15 years now, and again have done an incredible job. And so with that, how about we give a hand to the leaders of these groups? Really amazing and oftentimes thankless work that goes in behind this. With that in mind, the other critical part of the group is having a board that is willing to step up to help with the leadership. And so I know the turnover is coming up. I would really encourage, whether you're a new family or you've been here since 1998, I hope you'll want to rise to the occasion and be members of the board, because that is really a huge part of what makes these groups so successful. I also want to thank Teresa and Ken for their wonderful talks, which in many ways have set the stage for my talk, so they've actually made my talk much easier in some ways, so thank you to both of them. How about a hand for both of them, too? It's really gratifying to see how we work as a team, and again, I think when I first got involved with this, really starting in 1995, I never could have imagined to be where I am now with the group, the families, the sort of the combination of my research, my seeing patients, but this has taken it to another step for me, and I know over these last 25 years I've grown a lot as a person, and I really, again, want to just tell you how much I am honored to be part of this community, so thank you. One little pitch, and that is, it's getting a little late in the game, but we are having another conference in San Diego, just outside of San Diego, the end of June. It will also be streamed, I believe, just like we have in previous conferences, but the door is open. If any of you are looking for a reason to have a holiday in the United States and beautiful California, you are always welcome. Whether it's you guys or the other groups, we're all one family is how I look at it. So my door is personally always open to all of you. All right. Those are sort of the logistics things. So what I want to do is present to you kind of an overview of the work that really started back in the late 90s when I was still in my training. Like I said, we'll take a break about halfway through. But please, at any time, feel free to stop. Just raise your hand so they can get you a microphone so that the stream can pick up your questions. So don't hesitate at any time to stop me and ask any questions you might have. All right, any questions so far? Okay, good. Now let's see if I can figure out how this works. Okay, so I'll have to put on the old man's glasses here so I can see what I'm reading. Just to kind of give you an overview of what I'm going to talk about today, I'm going to tell you about the particular heart defect that I've been most interested in, called hypoplastic left heart syndrome. And I'm going to tell you how we've gone about finding the gene or genes and the types of cells that we think are what cause the heart defects that many of your children have. I'll also later on tell you about potentially a whole new different genetic cause for Jacobsen syndrome. And what has always been part of my vision is to hopefully apply what we learn from your children to the general population of children that are born with heart defects. And many of you probably don't know that by far, heart defects are the most common type of human birth defect. So about, almost depending upon how you define it, about 1 in 100 babies are born with some kind of a congenital heart defect. And believe it or not, even to this day, in most cases, we don't understand the cause or how it happens. And lastly, I'm going to share with you very briefly a story of a new gene that we've discovered. It has nothing to do with Jacobsen syndrome, but it shows how we can take advantage of our newest technologies to find genes to help us understand some of these defects. In this case, a defect of the aortic valve. And in adults, aortic valve surgery is the most common heart surgery, only behind coronary artery disease. And just for fun, some of these questions have come up over the years. In particular, why do some of your children have a heart defect and maybe another child with the same deletion has a completely normal heart? We're beginning to understand, we think, why that might be. And also, why is it that those of your children that do have a heart defect, why is that so variable? So in one case, it might be just a relatively simple hole in the heart. And in other cases, like I'll show you in a moment, the whole left side of the heart never develops properly. And just for fun, a lot of people have noticed that it seems like many of your children have red hair. And I actually posted this recently on one of the Facebook pages, and I got more response to that than any other topic I think I've covered. I didn't realize it was such a big deal, but we have some thoughts about why that might be as well. So I think, as you've heard from some of the other talks, when I got involved with this in the late 90s, there were three major, well, I didn't even know that until starting, but there were three major causes of mortality in people with Jacobsen syndrome. Traditionally, heart problems, bleeding problems, and infections. And I know, as you heard from Ken's beautiful talk this morning, nobody in theory with Jacobsen syndrome should be dying these days from infections. Do you agree, Ken? Yes, thank you. By the way, I did go to the immunology classes in medical school. I was one of the few. Bleeding problems, it still amazes me. Every so often, someone writes on the Facebook page, do any of your children have problems with bleeding? Well, we've known this for many years, and thanks to the protocol that all of you should hopefully be familiar with, similarly, no child or adult with Jacobsen syndrome should suffer any catastrophic bleeding. Teresa, do you agree with me on that one? Are you awake? Got you. I said, because of what we understand now about bleeding problems, nobody with Jacobsen syndrome should ever suffer a fatal bleeding complication. We hope, but we know that should be preventable. So really what that leaves us with is what I focus on, and that is the cardiac issues, because just in the United States in this last year, we lost 3 babies with Jacobsen syndrome and hypoplastic left heart syndrome. So that continues to motivate me to see if we can do a better job. So what I wanted to do is show you, some of you have probably seen this before, a cartoon of what a normal heart looks like. So this is a normal heart, left ventricle, right atrium, left atrium. So the left ventricle is the main pumping chamber of the heart, right? And that pumps blood out, the biggest blood vessel in the body, called the aorta, to get blood out to the body. And then when blood comes back from the lungs, after the right ventricle, it's the right ventricle that pumps blood to the lungs to get oxygen. That blood then goes to the left ventricle to get pumped out. The blood that goes to the body comes back to the right atrium, so that's what we call blue blood, and then to the lungs. So that's a normal heart anatomy and physiology. In hypoplastic left heart syndrome, what you see is a very, very small left ventricle chamber compared to that. And I will go into details in the next slide. But I think you can appreciate when it's this small, it can't possibly pump out enough blood. And that is why immediately after these babies are born, they will all die within days unless we intervene. And what you may not appreciate also is the valves on the left side of the heart. This is called the mitral valve, and that's called the aortic valve. Normally, they open like this, let blood go out, and close like that. In this case, the valves can be so severe that either they don't open properly or they may be completely blocked off. And then what you can also appreciate is compared to this nice big red aorta, the aorta here can be very small. So this is a very, very severe heart defect. And you could imagine, as we show here, that part of the problem may be that where you have this nice relatively thin wall, here you have this massively thickened wall. And you could imagine that maybe it's small because you have too much growth inward to sort of obscure that chamber. Does that make sense? All right, now my next slide I'm going to show you is actually of some real human hearts with hypoplastic left heart. If you don't like to see human hearts, close your eyes. I'll tell you when you can open. And it actually turns out that it's much more complicated than that picture I just showed you. So these are 4 different types of hypoplastic left heart. And just from a research standpoint, or I should say from a clinical standpoint, they all have a very small left ventricle chamber. And I'll show you in a moment that for now, the surgery that we do on these hearts is all the same, believe it or not. But if you look at, for example, this heart, so this is what was supposed to be that left ventricle wall or chamber, and you see this white scar tissue. But then what you also see is this massively thickened wall, like it grew into that, just like I showed you in that cartoon. So in this case, you could argue that that actually is because there's too much growth. So actually, one of my colleagues in San Diego thinks we should maybe call this hyperplastic left heart syndrome because of too much growth. Okay? And I agree with that. And when you look at this, under the microscope, you just have a ton of heart cells, too many heart cells. But now I want to show you what I think is almost the complete opposite of this variation. This is what happens in the 5 to 10% of children with Jacobsen syndrome, with hypoplastic left heart syndrome, in most cases. And here you see something very different from this. Here you have this very small slit. That was supposed to be the left ventricular chamber. So again, no way that's going to pump blood out. And basically what you have here, instead of this massive thing, is just this what we call a slit-like left ventricle. So in this case, you could think that maybe it's not too much growth inward, but it's actually a failure to grow outward to keep up with the rest of the growing heart. And I can tell you, nobody has had an opportunity to study any of these types of hypoplastic left heart syndrome the way that we can, now that we know, now that we know that for one specific genetic cause, Jacobsen syndrome, this is the most common type. So that's an incredibly powerful opportunity to learn some things about hypoplastic left heart syndrome that have never been discovered before. Does that make sense to everybody? And again, what I'm showing you here is a big reason why there has been such little progress in this field up until now. And this is another pretty graphic picture. This is a very sad story of a little baby who was born last June in Dallas with Jacobsen syndrome. She was diagnosed prenatally, and incredibly, the family reached out to me knowing that they weren't going to be offered surgery for this in Dallas. And this is her heart, same thing that I just showed you. And I literally flew down to Dallas after she passed away because this family offered to donate their baby's heart. So this is Sophia's heart, and we are, as I speak, doing studies to learn things about this heart that have never been performed before. And in fact, two other babies, one 6 days later and one a few months later, in the United States also passed away with Jacobsen syndrome and a hypoplastic left heart. And similarly, those two families also have donated their hearts to us. So now we have 3 Jacobsen syndrome hypoplastic left hearts that give us an incredible opportunity to learn what has never been studied before. So this is in a way the worst case scenario, but also an incredible example of heroics from families who during their most difficult time have stepped up to do something to want to help others, and I think that's incredible. Question? Which language? Oh, thank you, okay. Or no, you need to.
Yeah. Is it working? Yeah?
You're good.
Okay, perfect. I just had a question. You mentioned that 3 cases of Jacobsen, they all passed away.
Correct.
Even though, at least in one of the cases, it was identified prenatally.
Correct.
So were there complications during surgery? Was surgery not possible? Why did this
happen? So this baby, because of all the other problems that go along with Jacobsen syndrome, this is very debatable. I'm just telling you what happened. She had a very, very large deletion, by the way. The surgeons in Dallas did not think it was ethical to proceed with surgery. That's very debatable. I'm just telling you what happened there. The other 2 babies are different. They actually, both of them did go through multiple surgeries, and I think we understand better now, at least in one case. One was actually in San Diego, why they ultimately did not survive their surgeries. It was really a combination. I can tell you the baby in San Diego, the combination of the fact that that heart would not grow, but also the combination of infections and bleeding problems. And quite honestly, I'm not going to go into details. Looking back, some things should have been done differently that honestly I had no control over. The other baby was in Pittsburgh, Pennsylvania, and also went through multiple surgeries. And it's kind of a similar situation where I think it was a combination of the heart and bleeding and infections that just created an impossible situation in that case. So it's when you already have a very complex heart defect, And then you have these other complicating factors. It's challenging. Yes, Teresa? You might want to get a microphone. Yeah.
I don't know what you are going to do with hearts, but I would like to suggest you something on genetics ground. Is to analyze and check if there is a homozygous loss in the heart. Homozygous
Well, I'm going to come back to
loss.
that point because let me come back to that point. I'll
Okay.
just cut to the chase. What you're referring to, we have evidence that in a different patient living with hypoplastic left heart syndrome and Jacobsen syndrome, the other gene from the other chromosome is completely shut down, so it is like a homozygous.
Yes.
I'll come back to that. Don't worry
Okay.
about that. So the answer is we've already done that on a different patient to show that can be a mechanism. Everybody good? Any other questions? Moving on. So just like what I said, we think in some cases it may be from too much growth, so maybe that should be hyperplastic. And in other cases, specifically including Jacobsen syndrome, it's a failure of growth. Up until the early 1980s, if you had a baby with hypoplastic left heart syndrome, maybe some of you have heard stories or had family members, there was no cure, there was no treatment. You were told to bring your baby home and the baby would die within a few days. In 1983, oops, sorry about that, Dr. Bill Norwood from Children's Hospital of Philadelphia published a pioneering paper that showed that there was no cure, but there was a way to possibly reconstruct the heart to allow it to function. And this is just a cartoon showing that surgical approach. And basically what the surgeries were, which are actually 3 surgeries that we do now over about the first 3 or 4 years, is convert the right ventricle to do the job that the left ventricle was supposed to do. What you see here is the right ventricle is now hooked up or connected to the reconstructed aorta. And that the blue blood that I told you before normally comes back to the right atrium is now directly connected to the lungs. So this is what's called the Norwood-Fontan approach. And as I said, whatever single ventricle heart defect you have, whatever type of hypoplastic left heart syndrome you have, this is what you get if you want to have any chance for survival. And it's been an incredible step forward, but still, now you're asking the right ventricle to do the work it was never really designed to do. And this was, again, back in the 1980s, and we now know that many of these patients are going to ultimately develop heart failure as they get up into the adult age, and sometimes even much younger. And in those cases, the only possible treatment for them is a heart transplant. But I can tell you, a heart transplant, as we say, you might cure one disease, but you create another disease. It is not an easy path. Did you have a question? No? Or are you just stretching? Oh, okay, just stretching. All right. And again, this is why this field has been so limited, because nobody has appreciated those differences that I showed you before. But I believe that once you have a better understanding of those mechanisms, eventually it's going to help us to take better care, and I am already sure of that. Because, for example, the baby in San Diego that passed away, if we had a better understanding of that heart's ability to grow, we would have done things differently. And I'm going to tell you how we can figure that out next time, hopefully. Any other questions before I go on? Okay. So many of you have seen this old slide that I like to show. This is from the first child that I ever took care of back in July of 1995 with Jacobsen syndrome. I was taking care of him because of his heart defect, which was like hypoplastic left heart syndrome. but I think you can appreciate this is something that was in his old chart showing that he had lost a part of chromosome 11. And in his chart was Dr. Jacobsen's original paper from 1973 reporting a family with multiple family members affected. So because of my interest in the heart defects, this in a way was like a gift from Mother Nature to tell us that there must be a gene or genes in this region that are the cause of that child's heart defect. Make sense? So for the last 25 years, we've been trying to figure out that whole story. All right, so as you know, even to this day, we were just talking this morning that starting back then, we still are trying to learn. Ken, Teresa, you can agree with me, I'm sure, that even now, there's always more to learn, and this was our first attempt, this paper that we published in 2004 on actually 110 cases. Up until that time, the biggest group of patients in one paper was 17, so it was really a big leap forward. And so just focusing now on the heart defects, a few things became apparent. One is about half of your children have some kind of a heart defect, even two children with the identical deletion, and I'm going to come back. Teresa already sort of hinted at why that might be. And interestingly, in terms of hypoplastic left heart syndrome, about 5 to 10% of babies with Jacobsen syndrome are born with hypoplastic left heart syndrome. And that, to my knowledge, is more frequent than about any other genetic syndrome. So just from the research standpoint, it was sort of a blessing that somehow I came across that first child to give me an opportunity to be able to get to where we are now. The other thing that's very interesting is many of the other common heart defects that I see every day at my hospital, many also occur in Jacobsen syndrome. And another interesting question is why, again, might one baby be born with hypoplastic left heart syndrome and another baby born with an atrial septal defect, a hole in the heart? We have some, I think, good answers to begin to address that. And again, why is it that the other half don't have any problem with their heart? So one of the technologies that we've been able to take advantage of that many of you are familiar with now is what we call the microarray. And instead of that very crude karyotype picture that I showed you before, this technology allows us to get much more details of exactly where the breakpoint is of the deletion. And with the Human Genome Project, we can now get a readout to know exactly what genes are affected from that deletion. And since we know that the deletions are different in size, we could begin to map out the region in that end of chromosome 11 where we think this gene is, or genes. So if this is, let's say, a child with a relatively small deletion with hypoplastic left heart syndrome, we actually have come across several other patients with Jacobsen syndrome features, including, let's say, hypoplastic left heart syndrome, that, as shown in red here, have an interstitial deletion. So that's what we mean. It's within the chromosome and not to the end. When it goes to the end, that's what we call terminal deletion. And if you believe the same gene that's the cause of this hypoplastic left heart syndrome is the same cause as in this, then that gives us a region of overlap. And from that, we could narrow what was starting with maybe an area of 150 genes down to an area of just, 7 genes. And so the first gene that jumped out at us that you've heard me talk about before is this gene called ETS1. And ETS1 is a gene that encodes what's called a transcription factor. transcription factors are genes that actually turn on and turn off a whole lot of other genes. And we know that transcription factors are very important for development of organs, including the heart. But we also know in other situations, transcription factors, if they turn on or off the wrong genes at the wrong time, lead to cancers. It's just all about space and timing. So what was already exciting to us was the fact that when we narrowed down this region, there were already at least some studies showing that ETS1 has a critical role in the development of the heart. So that's where we've spent most of our time over the years, using animal model systems to learn as much as we can about this gene. But I'm also going to tell you about something very new and very exciting, and that is we now suspect that this second gene, which is right next to ETS1, called FLY1, which turns out to also be a transcription factor, and is actually very similar and related to ETS1. And we think that FLY1 actually may be the cause specifically of hypoplastic left heart syndrome in the human patients. Although we know that ETS1 has an important role, at least for the other heart defects that occur, and maybe it's actually the two of them together that almost always happens in the human situation to give you hypoplastic left heart syndrome. Does that make sense so far? Okay, now one of the things that sort of helped us to strategize how to move forward was that there was a patient from several years ago who had kind of like hypoplastic left heart syndrome, but not exactly like what I showed you. And that patient was found to have just a mutation in the ETS1 gene. Neither parent carried it, so it made it seem very likely that just that mutation in that gene could cause that type of heart defect, similar but not exactly like what I've shown you. And so, again, that further supported our hypothesis for why we think ETS1 is such an important gene. But more recently, what we have come across is actually a human patient who has two major problems. This is at my hospital. Paris-Trousseau syndrome, so the bleeding problem with the platelets, and hypoplastic left heart syndrome. And guess what anatomic subtype of hypoplastic left heart syndrome he has? Exactly like what we see most commonly in Jacobsen syndrome. So very recently now, we are not changing, but we are kind of shifting our direction a little bit to also study everything we can about FLY1 in heart development. So for now, I want to tell you more about what we've been doing with ETS1. And these are pictures of mouse embryos. And the first question you want to know is, where is this gene or the transcription factor actually functioning as the heart is developing? And don't worry about the details, but everywhere you see blue in these sections through a mouse embryonic heart is where the S1 gene is being expressed. And what we found is there are actually two types of cells that we know are critical for the developing heart in mouse or humans or even a fruit fly in some cases. And one is called the neural crest, and the other is called the endocardium, or actually cardiac endothelium. And just to tell you a little bit about cardiac endothelium, I'll come back to this, but cardiac endothelial cells are what make up the walls of blood vessels, okay? And in the heart, as I'll show you in a few minutes, there are specialized endothelial cells that have some functions, critical functions, in the heart. All right, so basically, what we think is that it's the loss of ETS1 in one or both of these populations of cells that gives us all these different heart defects. Does that make sense? But what's interesting, though, is all those pictures I showed you about the heart muscle either being overgrown or not growing enough, these transcription factors, ETS1 and FLY1, are actually not expressed in those heart muscle cells. But we know that there are signals from these other cells that are required for heart muscle to grow, and I'll come back to that. So I want to tell you about a really interesting population of cells called the cardiac neural crest. This population of cells was originally discovered in the 1970s by a lady named Peggy Kirby. And she was interested in learning how nerves regulate heart function. And what she discovered is that in the developing spinal cord or neural tube, there is a population of cells called the neural crest, or specifically the cardiac neural crest, such that if you eliminate these cells, you see heart defects. And the reason for that is because, as shown here, normally, as the heart is developing, these neural crest cells leave this developing neural tube and they travel or migrate to these different forming structures of the heart. So down here, these cells help to separate the two outflow tracts, so that's kind of the walls that separate the aorta and the lung or pulmonary arteries. Actually, even the wall that separates the two ventricles. So the wall that separates the ventricles, the wall that separates the two great arteries, and further on in those main blood vessels, okay? So what Peggy Kirby showed is if you disrupt this process, you get heart defects. And interestingly, in hypoplastic left heart syndrome, there is some evidence that part of the mechanism is due to a neural crest problem. Oops, sorry. And in this case, there is a neurologic disorder of the gastrointestinal tract called Hirschsprung's disease that is due to neural crest defects that happens far more frequently, over 100 times more frequently in people, babies born with hypoplastic left heart syndrome. So that already had kind of biased me to think that hypoplastic left heart syndrome must be, at least in part, due to some problem with neural crest cells. So my lab uses genetically engineered mice. And what we can actually do is specifically eliminate one gene, either in all of the developing tissues of the animal, or, for example, specifically just in a population of cells like the neural crest. And what you see here is this is a three-dimensional reconstruction of a mouse embryonic normal heart. This is done using a cardiac MRI, just like what we have had in many of us as adults, older adults. And what you see here is the left ventricle, the right ventricle, the red is the aorta, the blue here is the pulmonary artery. And you see here the wall separating the two ventricles. Can you see that okay? Or should we turn down the lights? Can everybody see that? Okay. And if you take like a section through that heart, this is actually the aortic valve, and what you see here, these three asterisks are the three normal leaflets, what we call a tri-leaflet aortic valve. And this is just another way of showing the same thing. And all this blue, or here all this hot pink, are actually neural crest cells. So we know that neural crest cells have a critical role, like I showed you in that cartoon, in the development of this aortic valve. When we get rid of the ETS1 gene in the neural crest cells, we don't see hypoplastic left heart syndrome. But we do see something else. We see a hole in the heart. So this is this connection here that you wouldn't normally see. So we call that a ventricular septal defect, or VSD. And we also see that this red vessel, the aorta, instead of normally hooked up to the left ventricle, is now pushed over and lined up with the right ventricle. And we actually call that double outlet because you have both the aorta and the pulmonary artery coming off of the right ventricle. That defect does also happen in Jacobsen syndrome. And if we look at that aortic valve, look at that, no blue. And here you only see 2 asterisks, meaning in about half of these mice, one of those leaflets is completely missing. So this again supports the hypothesis that neural crest cells have a critical role in the development of these structures in the heart. So namely, that wall and also the developing aortic valve. And here, compared to that, I think you can appreciate there are far fewer pink cells. So just hang on to that thought, because I'm going to come back to that towards the end of my talk about a whole other story of neural crest cells. All right. And remember that cartoon I showed you with the cells kind of moving down? Of course, that was just one snapshot. But actually, what we can do is label these cells. So this is actually, if you see these arrows here, these are neural crest cells in a developing mouse embryo. I think you can see this is where that neural tube was. Okay, that's the neural tube. And these are the cells that are leaving the neural tube to migrate down to what is the developing heart. And what you can see here, thanks to the person in my lab who did these beautiful experiments, Lizu Lin, is this really nice kind of stream of cells moving to where they need to go. So that's a normal mouse heart. And when we delete our Jacobsen syndrome ETS1 gene in that same, in neural crest cells, we see a complete loss of that normal traveling or streaming of neural crest cells. So basically, what that shows is that this population of neural crest cells never gets there from the neural tube to form that wall and to help in the development of the valve, the aortic valve. Okay? And to answer Teresa's question, what we could actually do from some of our living patients is actually grow cells from a patient with Jacobsen syndrome and a hypoplastic left heart syndrome. And I'm going to show you a video about how we do that in a few minutes. But what you can do is actually take skin cells, or now these days blood cells, and reprogram with some genetic magic those cells to be heart cells, or in this case, neural crest cells. And we did that, in this case, from 3 normal controls. So don't worry about the numbers, but this just shows you 1.0 is normal expression of the ETS1 gene. And these 2 are 2 different patients with Jacobsen syndrome and hypoplastic left heart syndrome with that same anatomic subtype. And in one of the 2 patients, there is no expression of ETS1, which means from the normal chromosome that's not affected, at least by deletion, for some reason, that gene is not being expressed as either. So like Teresa said, effectively that's like a complete knockout, which is actually what I just showed you in the mouse. So you get rid of both copies. So that's one explanation, but interestingly, the second patient, we did the same experiment. There is expression of ETS1 from the other chromosome, although we can't be sure. We haven't studied this any further, but we think that this copy of the gene may not be functioning normally. So effectively, you have a significant loss of function of ETS1 completely here, and to some extent there, that causes the problem. Okay, so I'm about halfway through, so why don't we take about a 5-minute break, catch our breath, stand up, stretch, drink some water, use the restroom. Does anybody have any questions? The next most major part of the rest of my talk is going to be coming back to how we think this causes hypoplastic left heart. Okay, so let's regroup in about 5 minutes. Sound good? All right, so most of the rest of my talk is going to be about the role of ETS1 in the other population of cells that I told you about called the cardiac endothelium. And in this cartoon, as I mentioned, endothelial cells are what make up the walls of blood vessels in the body. And specifically in the heart, you can see here this green lining. That is a special population of endothelial cells that make up the inner lining of the heart. That's called the endocardium. And it turns out what we know already before we even got into this is the endocardial cells secrete critical factors that are required for the development of the ventricle. And we'll come back to that theme in a little while. The other place where you see ETS1 expressed in the heart endothelial cells is actually in the branches of the coronary arteries. So that's called the coronary vascular endothelium that actually supply blood to this outer part of the heart muscle. Okay? And again, we know that these very special coronary vascular endothelial cells also secrete factors that are critical for the developing ventricle. So you could certainly hypothesize that if you take away ETS1 in either the endocardium or in the vascular endothelial cells, that might cause maybe even hypoplastic left heart. So what I'm going to show you next is actually a paper that was published really before we even identified ETS1. Oh, this isn't working now. Where did, come back. For some reason, neither function is allowing me to advance the slide. Did you break it? I can figure out hearts, but I can't figure out a project. Yeah, it's not responding. Did it freeze or something?
No, maybe no.
Oh, yeah, okay. What did you do? What's that?
No.
No, you hit advance.
So this,
Okay, so just do that. All right. So it turns out, some of you may have seen this slide, that many of the same genes that are involved in the development of the human heart are actually involved in the development of the heart of something you see in your kitchen, what's called a fruit fly or Drosophila. And I am very, very fortunate to collaborate with some people in San Diego that study fruit fly heart development. And interestingly, again, before we discovered the ETS1 gene, this paper was published showing what happens if you get rid of the ETS1 version in the fruit fly. And this, if you can see this, is just a little tube. So each one of these green and yellow is a heart cell. So this is a fruit fly embryonic heart, which is basically just a tube. And this group published 20 plus years ago that if you get rid of what's called pointed, so that's the fruit fly version of ETS1, you see, in this case, actually this overgrowth of cells, of both green and yellow cells, that pretty much obliterates that lumen or chamber. So of course, these guys had no idea what hypoplastic left heart is, but I thought that that was a very powerful observation that suggests that maybe pointed or ETS1 is doing something to regulate the growth and development of these developing heart cells. Okay? Now that's a fruit fly, not anywhere close to a human, but I can tell you my collaborator has identified another gene called Tin Man that has led us to identify human patients with a mutation in that gene that alerted us to have to put in a pacemaker that has literally saved the lives of a number of our human heart patients, thanks to a fruit fly. So don't swat a fruit fly next time you see one, because they actually are life-saving. All right. Next slide. So one of the other animal model systems that we use is a frog. And I am very blessed to have another collaborator named Shui Ni, who works on frog heart development. She's a PhD. And a frog heart normally just has one ventricle, and this is what I'm showing you here. And the green are just all the heart cells, the normal heart cells. So this is a normal frog ventricle. And she did an experiment similarly to eliminate ETS1 in the precursor cells that give rise to the cardiac endothelial cells. And look what you get here. This is a much smaller heart, and I can tell you they can do video filming, and this heart does not beat well at all. So it's small and does not function well. And those are really two of the hallmarks of hypoplastic left heart syndrome in humans. So in frog, this is really exciting because this actually makes us think that ETS1 is the gene that can cause hypoplastic left heart syndrome. Well, of course, nothing is as simple as what you would like, because now we turn to the mouse model, because the mouse heart is much more like a human heart. And I think, like I showed you before, this is actually a normal mouse heart just staining all the heart cells. So this is the left ventricle, this is the right ventricle. And if you enlarge that, you see here, actually there's this part, this is kind of the outer layer of muscle. That's actually called the compact zone because it's more compacted. And then you see these kind of like finger-like projections. That's another population of heart cells called the trabecular layer. Okay? And I can tell you to this day, when we knock out ETS1 in the cardiac endothelium, we've never seen hypoplastic left heart. Damn, right? So that's kept me up quite a lot at night. But we do see something that is also very abnormal, though, with the ventricle. I think you can appreciate here, this compact zone, completely gone when we knock it out in the cardiac endothelium. And what we see is just sort of this very lacy appearance. And this mouse actually didn't even make it to birth, because you could assume that this heart could not possibly do what this is supposed to be doing. Okay, so yes, it's exciting. We have evidence to indicate that ETS1 has some role in the normal development of the ventricles, but we're not seeing hypoplastic left heart syndrome. So why is that? Let me go on and share you some additional information. So Lu Wang in my lab is a PhD, originally a postdoctoral fellow, and now a project scientist. And one of the things he could actually do is study those mouse hearts where ETS1 is eliminated, and he could actually identify those genes that ETS1 regulates. Remember I told you transcription factors turn on and turn off different genes? So that's what allows us to begin to define what we call these genetic pathways. And what you see here are some of the genes that ETS1 directly regulates, okay? And if you look at their functions, as you would have hoped, basically they're all genes that are involved in the development of the cardiovascular system. So that's a very reassuring result that we're hopefully on the right track. And very interestingly, if you look at some of these genes that ETS1 directly turns on and turns off, several of these direct downstream targets, as we call it, what's the problem? My wife has never told me that. All right, I have to talk loud. Sorry about that. I'll wait till he gets back. Okay, so what Lou was able to do is identify some of these direct downstream, oh. Then I'll start singing. I have fantasy. Hang on. I think it's kind of too top heavy, I think. All right, I'll just hold it, that's fine. Yeah. All right, here we go, all right. Is this okay? Okay, thank you, Sandra. You've probably never told a guy to speak louder before, have you? Okay, so when you look at these downstream target genes, what's really interesting is, for example, this gene has actually by itself, when mutated, been associated in, or been found to be mutated in patients with something called tetralogy of Fallot. It's another heart defect that does occur in Jacobsen syndrome, but not as common. Whereas this gene called Claudin-5 has actually been shown in the frog to cause that same defect of the ventricle that the ETS1 knockdown causes in the frog. So what we think is happening, so for example, I know I can share this first gene called ALK1 has been associated with something called total anomalous pulmonary veins. The veins, instead of going to the left side of the heart, go to the right side of the heart. For example, one of our US families, a child was born, there are a couple of kids that have been born with that. So what we think might be going on here is that for any one patient, whichever gene on this list is the most sensitive to the loss of ETS1 will determine what heart defect, if any, you get. Does that make sense? So I think for the first time, this gives us an ability to answer that question I asked before. Why do some of your kids have different heart defects? Or why do some kids not have any heart defect? So it's either the complete loss of the one copy or an abnormal version of that one copy combined with any one individual's sensitivity among these genes to the loss of ETS1. And just like different people have different eye color, different people have probably different relative sensitivities among these genes to the loss of ETS1. Does that make sense? I hope. So, just to summarize here, talk about the rarest of the rarest, right? So these are all different patients with Jacobsen syndrome over the years. Edgar is actually the first kid whose chromosomes I showed you back from 1995. This little girl had a much smaller deletion, but with hypoplastic left heart syndrome. This little baby was also born with hypoplastic left heart syndrome, but with a very small deletion. And these are actually 2 of the 3 babies from this past year that I told you about. All right. Last few minutes, I'm just going to show you a few other things that we've been doing that are kind of offshoots of Jacobsen syndrome. This is actually literally just from a couple of months ago. I was taking care of a patient in the hospital, a 15-year-old girl, and here's what she has: hypoplastic left heart syndrome with the same exact anatomic subtype that happens in Jacobsen syndrome, persistently low platelets, persistently low white blood cells with low antibody titers, growth hormone deficiency, mild intellectual disability, and some very mild facial features, including the down slanting eyes. Believe it or not, I had never seen her until a couple months ago. She had never had genetic testing. Would anybody in this room like to make a guess as to what syndrome she might have? Well, I made a guess, and guess what? I was wrong. Maybe. Because it looks like, so we did the microarray, and guess what? No deletion. What? I said, can you please go back? I've asked them now 3 times. I copied Teresa in my most recent email. So then the next question is, what about maybe a mutation in ETS1 or FLY1? Nope. But interestingly, what was found is a mutation in a different gene on a different chromosome. And this gene is involved in something that Teresa touched upon a little bit called epigenetic regulation. It's a whole nother level of how genes can be regulated. And in this case, what this gene does is it actually makes some chemical changes in the DNA that can normally either shut down or turn on the expression of genes through what's called methylation, so adding a methyl group. The good news is we actually now, I've made a connection just in the last few weeks with the world's expert studying this gene. And he's going to do an analysis on this patient's DNA to determine if, in fact, genes like ETS1 or FLY1 or maybe something else in 11q might be affected because of the loss of this gene. So that's a very new, novel story. As I said before, it's always humbling to me how just when you think you know everything, you don't. So it's like the older we get, the dumber we are, the more ignorant we are. Anyway, it's an incredible story. So another story I want to show you is what I referred to before, how we can look at patients and take cells from a patient and reprogram them. And this is just a cartoon showing you a company that I've started to work with that can do exactly that. So they take skin cells, they reprogram them, as you'll see here, and bottom line is they can do some really incredible studies with reprogrammed heart cells from these patients. And then they can actually create, as I'm going to show you in a minute, basically what looks like a beating heart from these patients, whatever patient you're interested in. Keep watching because this gets really pretty cool, I think. So that's basically just showing a single heart cell by itself in a test tube beating, but they can actually make heart-like structures, heart tissue, from millions of these cells that are, again, derived, let's say, from a patient with Jacobsen syndrome and hypoplastic left heart syndrome, using their magic. And this is what's called a tissue strip, so it's like just that, a strip of heart tissue that they've engineered. And look what they can do. If you watch this carefully, it actually contracts. And the way they have this all set up is you can actually measure the function of that tissue. And taking it one step further, they can actually engineer a pumping heart chamber. If you look carefully here, keep watching. Look at this. This thing actually pumps and actually can eject a volume that can be measured. So why is this so important? Well, remember in the beginning of my talk, I told you that my patients with hypoplastic left heart syndrome, they have the 3 surgeries. The first patient that I ever diagnosed is like 27 years old now, doing great. But some of my other patients don't do great at all. And some of those patients don't survive more than a few years before they either die or need a heart transplant. And the problem is, in most cases, we can't determine which patient is going to follow which clinical course. Using this technology, we think that from the beginning, we can perform this analysis to help us to predict from the beginning who's at risk for the development of early heart failure, or like the baby that died in San Diego, which patient has a better ability for their heart to maybe grow so you could still have 2 ventricles, because sometimes you really can't tell just by looking at it. And so in the future, we think that adapting this technology to hypoplastic left heart patients will allow us to very proactively personalize our care that will ultimately result in much better outcomes than, let's say, those 2 babies I showed you who really languished for 9 months. I think if we had this technology applied to them, I think one and maybe even both of them would be alive today because we would have known better what to do. And if you think about one patient alone, the baby in San Diego, he lived to about 9 months. I don't know specifically, but he probably had a medical bill of over $2 to $3 million. So imagine, this is a little bit expensive up front, but even if you could prevent one patient like that, it's going to pay for itself 1,000 times over. Okay, I'm going to skip this slide because that basically lays out what I just told you. Here's just, again, another image of that beating heart chamber. As you can tell, I think this is pretty cool. In fact, the guys that started this company, I told them, if you had told me 15 years ago that you could do this, I would say you're absolutely crazy. So these are PhD bioengineers who have spun off from academia to develop these kinds of technologies that, combined with people like me, academic researchers, we can really work together and collaborate to be able to do things that, you know, none of us could do individually. And I think that's an incredible statement about how academia and academic spin-off biotech companies can really join forces to do some amazing things, in my opinion. Yes, sir. Yeah, this is about 1 centimeter in diameter. Yeah, half an inch. Yeah. I mean, you know, there are obviously limitations, but it is a functioning thing. And actually, the first set of experiments we're going to do, thanks to a collaboration I have with the Mayo Clinic, is to do this from a couple of patients that have developed very early right heart failure as sort of a proof of concept that we can reproduce using that or the other applications what we know happens clinically. I'm very confident we will, and if so, then I think we're really onto something. Okay, so he is asking a really good question. Is it possible to let this grow by itself? In this situation, the answer is no, because this is really just pure cardiac myocyte pumping cells. However, there is an emerging technology that Ken and Teresa probably are familiar with called organoids, and in this case, cardiac organoids. And what some really smart people are doing, this is relatively crude, right? It's pretty simple. It's just pumping heart cells. But people are actually taking stem cells, and again, with the same kind of trickery, they're actually inducing these stem cells to grow into organs. So that's what we call organoids, including the heart. And now that you ask this question, because it's so cool, we're actually I have a new collaborator who just moved to San Diego from a very well-established lab up in San Francisco. And we're gonna work together to take, again, some of these patients, like the patient with hypoplastic left heart syndrome and the FLY1 mutation, and he's gonna apply his technologies to generate a cardiac organoid in a petri dish, and I'm sure something's gonna be really messed up with that heart. In that case, since we know the single gene mutation that's the cause, what we can do is then come back and use something called CRISPR technology. We can actually go back and repair that mutation to make it normal, and then make a cardiac organoid out of that. So in a way, that becomes the control to show that it's really that FLY1 gene mutation that's the cause of the abnormal heart development. I'm sure that was a much longer answer than what you expected to get from me. Great question, though. So definitely some limitations, but I think if you ask the right questions, this can give you some very meaningful information, including, for example, are the cells from like a Jacobsen syndrome patient capable of growing, right? Because sometimes we get a mildly hypoplastic ventricle, not as bad as what I showed you. And that's actually what happened with that one baby in San Diego. And, you know, my surgeon wanted to do things his way, but it was okay. I know what he based it on, and I didn't totally object to it. And he aimed for what we call a 2-ventricle repair, and it didn't work out. But for that patient, I think if we had done this and done assays to show that those heart cells will not grow over time, we would have gone down the single ventricle route. All right. And just to wrap up, this is actually an echocardiogram from a fetus that we came across a few years ago. Oops, sorry about that. This is the right ventricle. This is the left ventricle. I think you can appreciate there's a little bit of a problem there, right? Just a little bit. Well, it turned out this fetus that was about 23 weeks gestation was completely missing their aortic valve. Now, remember what I told you. Every time the left ventricle pumps blood out the aorta, normally the valve opens and then it has to close so blood moves forward. In this case, every time the ventricle pumps blood forward, it just leaks right back. And that's why you got this humongous left ventricle, and this fetus was not viable, so the baby died a few days later. However, we were able to get DNA from this fetus, and we actually found a new disease-causing mutation in a gene that had been already shown to have a critical role in preserving cardiac neural crest cells. Now remember I showed you before in those images, neural crest cells, those blue or the pink cells, are in the aortic valve. So in this case, what we think is that because of this mutation, those cells that are supposed to go to the developing aortic valve were killed off basically because this gene actually prevents cells from dying. We call it a tumor suppressor cell. So I have a brand new postdoc in my lab now who's going to study this gene to learn more about what this gene does to regulate cardiac neural crest cells during heart development. So, you can see how I can tell you I never really would have had this insight if it weren't for the work that we've been doing with the Jacobsen syndrome story and neural crest to come up with this hypothesis. All right, so I've kind of already talked about this. Why do some people with Jacobsen syndrome have heart defects but not others? I think I kind of went through that. What is the basis for the different heart defects? I covered that. But last and not least is why do some of your children have red hair, right? Now, someone corrected me on Facebook when I posted this several weeks ago. What I'm going to argue is there are many causes genetically of red hair, right? There are plenty of people with Jacobsen syndrome whose parents have red hair. So it's not because of Jacobsen syndrome in that case. But we do know that the ETS1 gene is also required for a different population of neural crest cells that are actually required for hair color. And if you delete it in those cells compared to a normal mouse, look what you see. It's not quite a red-haired mouse, but you certainly see these patches of loss of pigment in the fur. And if you look carefully, this poor little mouse has a very severe spinal defect called kyphosis, which is kind of like scoliosis. And if you look really carefully, this little mouse has a swollen head due to something called hydrocephalus. So anyway, at the end of the day, this might be one contributing factor as to why many of your children seem to have a much higher than predicted frequency of red hair. And I think that is really the end of my talk. I really do want to acknowledge some incredibly talented people that I work with either directly in my lab. This is Aiden, who's an undergrad student applying for medical school. This is Lizu Lin, who I told you is the incredibly talented senior staff scientist who did all those incredible studies on the neural crest cells. This is Annalyn Schroeder, who has been working with my collaborator on the fruit fly heart development and the role of the ETS1 gene in fruit fly heart development. This is Lu Wang, who is now a project scientist in my lab, who is an incredibly talented and brilliant scientist, arguably the best I've had in my lab in 25 years, who has really opened up my whole understanding how we think these genes in 11q are causing hypoplastic left heart syndrome, and that it's not just a neural crest component. And so perfect timing, Lu was actually a friend of Lizu's from back in China, and Lu got his PhD on vascular endothelial biology, so he was a perfect match for my lab. And this is Antonella Pinto, at the time a postdoctoral fellow in another lab down the street from me, who did all those human neural crest cell studies that I showed you. And then just to finish, I'm incredibly blessed, and I think this is a really important point, that you've heard me kind of mention what some of the problems have been in studying this complex disease or set of disorders. And what I'm really proud of is that I've assembled a group myself. This is Karen Okor and Rolf Bodmer. They are a husband-wife team who work on fruit fly heart development. So that's Annalyn's boss from the slide before. They are incredibly talented PhDs and really good friends of mine. Rolf is originally from Switzerland. And so they are very hungry to study not just heart development, but how it applies to human disease. And I can tell you, honestly, I have a number of my patients walking around that are only alive today because we knew about the gene that they have a mutation in that these guys discovered. And I should have shown you a slide when I actually got to introduce them to one of the families. I mean, it was really powerful. Lou, I already told you, who is now graduated, still in my lab, is now a staff scientist. I can't imagine my future without him in my lab because he is so talented. And then Shui Nai is a PhD embryologist who studies heart development in the frog. So those pictures I showed you is all from her work. And lastly, just for a little reminiscing, this is actually the first baby I diagnosed with hypoplastic left heart syndrome in 1996. Who's obviously doing really well, thank goodness, because of those advances. But what is sobering is, I think as I've made you appreciate, her future is very uncertain. And that's what motivates me to want to keep this going. So I am done, and I would be happy to answer any more questions or give you a pillow to go to sleep after such a long talk. Thank you. That was a lot of stuff. But again, I just want to come back and say, you know, this group, what you guys have done to help me, including recently a nice contribution to help support my research in my lab and to help support people like Lou, is just, you know, overwhelmingly generous. And I can't thank you and the other groups, the Spain group and the American group, for all the support over the years. So again, it's not possible without you guys. Any questions? Nope. Beer time? No, there's some more activities. All right, thanks again.